Thank you for the introduction, and thank you for having me and giving me this opportunity to present my work. So today I'm going to be talking sort of generally about work I've done using experimental evolution to study variation in life history patterns. So just a quick outline of the talk. I'll give a little bit of background, talk about some of the phenotypes, like what do I actually mean when I'm talking about variation in life history patterns. And then I'm going to be talking about some recent work I've been doing using metabolomics to understand what we call this accelerated aging phenotypes. I will talk about some genetic stuff in there, so it's still appropriate for the venue. Um, okay, but sort of broadly, a lot of my work focuses on using experimental evolution. So I just wanted to quickly, talk about what is experimental evolution, why should you care, kind of thing. So the general idea behind experimental evolution is you bring organisms, usually some sort of model organism, into the laboratory conditions. You choose some selective pressure, some challenge. So here, we can, unlike my little cartoon, we can say we're putting something in their food, right? So these populations now have a different food source, and then we're letting them adapt to those conditions for many generations. Then we have, the final form of the controls, or not necessarily final form, but control and adapted populations. Then either through comparing those two groups or seeing how phenotypes have changed over time, people have used these experiments to learn a lot about general evolutionary dynamics and thinking in terms of like, what's the rate of adaptation, how fast do traits change, and then you can perhaps get some insight into what are the mechanisms underlying what type of change you're seeing. For instance, if you're doing adaptation to a different diet, you could look at all the phenotypes involved in that and see, okay, what does it actually mean to be more adapted to this diet? I guess maybe a little over 10 years ago, people started combining experimental evolution with DNA data, so genomic data, in what are called evolve and resequence experiments. And you're essentially, adding to this framework where you're saying, okay, before we would see how these phenotypes are changing and so on. Now we can see what genetic changes are underlying those phenotypic shifts and, answer just general population genetic questions about these dynamics, quantitative genetic questions, or just thinking broadly about what's the genetic basis of the trait you're studying, if that's what you're interested in. All right. So there's that sort of combination of dynamics or getting a better functional understanding of whatever trait you're interested in. So broadly, this is split into sort of two types of experiments, right? So you have experiments with asexual systems. So typically, you're going to be E. coli and yeast, where we have adaptation being fueled by new mutations that are going to, sweep to fixation. So your classic selective sweep. And then we have sexual systems. This is mostly Drosophila, some yeast work, like folks like Molly, Tony, and I have tried out. And here, all of the evidence points towards adaptation being more from standing genetic variation. And oftentimes we don't even really see fixation, right? We might just see shifts in allele frequencies versus sort of these more sweepy patterns. And I would say my work exclusively focuses on these sexual dynamics. So that's what I'm going to be talking about today. And I would make the case based on what we've seen in the literature on this topic, even when we're getting out to hundreds of generations or even larger population size with some of these yeast experiments, we're still seeing most adaptation is involving standing genetic variation. So I would argue these are very, very relevant dynamics to real populations that are evolving that we would care about on human relevant timescales, right? So I don't think it all has to be about mutations. So in one of the systems, like Christian mentioned, I've done a lot of work with and still continue to work with that we're sort of moving into my lab, was created to study the evolutionary theory of aging, right? So answering this question, why do organisms age and what controls the rate at which they age? And note when I say age, I mean senescence, right? So the actual physiological decline that occurs over time. So I just briefly wanted to go over this figure to kind of orient everyone to what is this evolutionary theory of aging that was used as the basis for this experiment. So the evolutionary theory of aging, let's see if I can get this pointer business working, is basically this idea, the strength of natural selection is strongest prior to the start of reproduction, then following the start of reproduction, the force of natural selection acting on an individual goes down, right? So if you wanted to think through this, you can imagine if you had like a lethal dominant mutation that had early acting effects, selection would be very, very efficient at removing that. Those individuals don't even get to make it to the point where they can reproduce. But if you had a mutation or a lethal mutation with late acting deleterious effects, it would persist in your population at higher rates than you would otherwise expect, because by the time its deleterious effects are felt, it's probably been passed on to the next generation, right? So the classic example of this would be something like Huntington's disease, which is a lethal dominant, but because of the age of onset being so late, it persists in populations at higher rates than we would expect, based on its mutation rate and all that. And then within this, and I'll kind of come back to this, there's sort of two hypotheses sort of underlying this evolutionary theory of aging. So there's the mutation accumulation hypothesis. This is saying this senescence or aging is because over time populations accumulate a bunch of these alleles with late-acting deleterious effects, and that's why, as things get older, they're essentially breaking down, right? Then we have the antagonistic pleiotropy hypothesis. So this is actually saying, as a result of these dynamics, if you had a mutation or an allele that had benefits early on in life, even if it had costs later on in life, it would still potentially be favored by natural selection because it's conferring benefits at the areas where natural selection was its strongest, right? So how would you sort of test this empirically? This is what sort of was the genesis of this system, where Michael Rose, back in, I think, the late 70s, was like, okay, how can I experimentally test this, right? So one idea is if you wanted to use selection to create populations with increased longevity, you would simply you try to delay the start of reproduction, right? So if you could artificially do that, you're extending the period of time where natural selection is acting at full force. It should be getting rid of those more and more of those deleterious alleles that have accumulated, right? In terms of doing this with flies, it's actually relatively easy to move the effective start of reproduction, right? Because oftentimes flies are being maintained on non-overlapping generation cycles. So the baseline populations for this system were maintained on a 14-day generation cycle. So only eggs laid on day 14 are used to found the next generation. So if you were to shift this outwards to a later date, you're effectively shifting the first age of reproduction, because that is the only day that's seen by natural selection. So this was done shifting this date all the way out to day 70, and then this was done over many generations. And then when you look at the survivorship curves for these populations, so in blue we have the baseline populations, and then in red we have these old selected flies. You can see it's shifted, meaning they, on average, are living much longer than the controls, right? So then years later in the 90s, Adam Chippendale had the idea of like, well, okay, if we could use this principle to select for increased longevity, we can sort of go in the opposite direction, and that should produce reduced longevity. So getting sort of both ends of the spectrum on this. And to do that right, you're just moving your collection day earlier. So again, going back to this fly system, they move all the way back to day 9 to day 10 to try to select for early reproduction, reduced longevity, accelerated development, however you want to characterize it, right? I will say here, there's kind of a hard limit where you can't really seem to get to 7 days. So at some point, you do run into just limits of development, right? And then when you look at that, you get the expected pattern where survivorship is shifted in the other direction. So these populations are not living quite as long anymore. Okay, so a lot of the populations I've sort of inherited or continuing to work with are focused on what are called the C-type populations. So these are flies on a 28-day generation cycle. So, pretty relaxed selection. They live relatively long period of time. And then we have these what are called A-type populations, which are on that sort of 9-day selection, or 9 to day 10 day collection window. So they have accelerated development. If you were to actually look at the relationship between these, like you would have like replicate A1, replicate C1, those share an ancestry. So we do have that pairing within the system that helps with some of the comparisons. Within the A types, we have 5 populations that at the time of the most recent work have around 550 generations of selection, and then 1,100 generations of selection, because we essentially recreated these populations in some work that I'm not really going to talk too much about today, looking at how repeatable evolution is if you go back to the source ancestor and try to recreate things. Does it proceed in the same way in a parallel fashion? And at this point, when we look at this system, we know we have very, very high, we usually see very parallel responses to selection. It's very rapid, and we get really high convergence. So these new A populations that have 550 basically look identical to these more longstanding counterparts, despite the fact they were created years apart, right? And then the Cs are reflecting the same pattern. So we have the Cs that are 200 and then 450 generations. So we consider this a system of like a 10-fold replicated system, right? We have 10 As, 10 Cs because of this, even though there's some differences in generation time. Let's see. Okay, so next I'm going to talk a little bit about some of the phenotypes to, fill out the landscape of like, because obviously, aging is a pretty complex phenotype, so you wouldn't simply expect like they live longer and nothing else happens. So what's underlying all of this? So when we're thinking about the phenotypes associated with accelerated, or what we call this accelerated aging phenotype, we have, selection for early reproduction has resulted in very distinct age-specific mortality patterns. So on the top here, we have the A populations, females and males, and then on the bottom you have the C. And you can see there are days where when you get to the end of the C lifespan, well before that, all of the A's are dead, right? So there's a very distinct difference in their longevity and survivorship patterns. We also see this reflected in a bunch of other traits, and we, thinking in terms of aging, we're all thinking of these different trade-offs that are allowing this to be possible. So when we look for selection for accelerated development, or we also, we find accelerated development, right? So on the top, we have the A's and the C's again. So this is looking at pupation rates, and this is hours from hatching. So you can see the A types are pupating much earlier than the C types. So that's also happening as part of this selection, but then they're living shorter lives, right? So there's some apparent trade-off happening there. Next, we also see fecundity seems to be almost calibrated to their reproductive windows, or that appears to be what's happening over time. So again, looking at age from hatching. In the A types, you get this initial ramp up that sort of peaks around the time we would actually be collecting eggs. So in that day 9 to 10 range is when they have their peak fecundity. And then you kind of plateau and decline after a while. Whereas with the C-type population, that increase in peak comes at a much later time. And part of the reason we don't have earlier fecundity data for the C-types is because they don't exist as adults as early, right? So there are no measurements to be taken. When we look at sort of a broader scale, so this is looking at these 2-day intervals, we can see in the right column, we ran statistical tests to see if you had differences in fecundity in these different windows. So there is like that period where their fecundities kind of overlap when the A's are going down and the C's are going up. But then after those periods, the C types always have higher fecundity, right? So there's this underlying trade-off in terms of how their reproductive patterns have shifted to be optimized around their reproductive windows. And then we also know this is correlated with different patterns of stress resistance, right? So when we look at cardiac arrest rate, this is some work from James Kezos, who was interested in, is this a good model for studying heart disease? He would run a current through the flies and then see if they go into cardiac arrest or if they were able to survive this. And then we find that the hearts for the C types are much more robust, so they don't go into this cardiac arrest as higher rates, even when shocked in this way. And then we also see patterns in starvation and desiccation resistance, right? So the sort of general story about this model for accelerated aging is they have shorter lifespan, faster development rates, and reduced stress resistance. So we think this is a very, complex phenotype, as you would imagine when you're talking about aging, and sort of inherent to it is these different types of trade-offs going on. So next, I'm kind of going to get into this metabolomics and accelerated aging. But before that, I wanted to actually talk a little bit about some of the genetic work I've done in this system, because that's kind of what's motivated me to start looking at these other avenues to try to understand functionally what's going on. So when we do thinking about like what is the genetic architecture of the accelerated aging phenotype, we could sequence them. So we sequence the C-type populations, we sequence the A-type populations, we use your CMH test to compare SNP frequencies between them to see like where are they different, right? And this is you can see here, just to orient you on, this is a Manhattan plot. So on the y-axis, you have negative log p-values. On the x-axis, you have the different chromosome arms. So you see there's widespread SNP differentiation across the entire genome in response to this selection. So we would say this is, apparently very highly polygenic selection or trait. Yeah. Okay. Next, we've also done some work, transcriptomic work, to seeing how this translates to patterns of gene expression. So here we looked at 2 days, day 14 and day 21. We initially chose day 14 because if you look at the mortality patterns, that's where they actually have pretty similar age-specific mortality rates. So we are thought was, okay, maybe they would be less differentiated at that point because they're more phenotypically similar. And then day 21 is when you have the sort of greatest difference in mortality rates between the two. So maybe highest difference. But in both cases, we find tons of differentially expressed genes. And this is based on whole bodies, like 200 females in each group. So next, we're so like, how do we make sense of this, right? So we have all of these SNPs, we have all of these differentially expressed genes. Is there some way to connect these two to think more about what's actually going on in this system in terms of the underlying genetics? So this is where we've done some work trying to link the genome to the transcriptome using a lasso variant. So this is called FLAM. So in terms of what a lasso variant is, it's basically a method for feature selection. So you're doing regression, but you have a penalty term. So as you continue adding variables, you're adding a penalty to that, and then you're shrinking uninformative variables to 0. So using this approach, you can say, like, let's say we have all of these SNPs we're interested in. We have all of these differentially expressed genes. We can identify which SNPs best predict the expression patterns of different sets of genes, right? So that's one way of linking the two. Obviously, you're making a big assumption there that predictive power is somehow translating to they're more likely to have a causal relationship. So there's a little bit of a game going on there, but we'll sort of just make that assumption for now and see what it tells us, right? So when we look at this, I will say when we use the SNPs, we didn't use all of the significantly differentiated SNPs. We used the windowed approach. So we would use the most significant SNP within 50 kb windows, and we tried a few other window sizes and have that as the number of predictors. And part of this was because obviously in this case, predictors are correlated because of linkage, right? So we wanted to try to account for that in some fashion. And this is doing that, linking it to the day 21 data, so you can see they're very highly connected, right? And just to look at one example of this, so we would have this specific site or this specific gene, and then these are all the SNPs that are the best predictors of the expression for this. And so if we look at the macro level, on average, a given differentially expressed genes will have 4.5 candidates that are good predictors of that gene, and a single SNP will be involved in 15 predictive responses. So if we take this to be true, it suggests that we have widespread pleiotropy in this system, right? Because you have this high interconnectivity between the two. And specifically with respects to aging and results we've observed in the past, where we don't really see a lot of fixation when these populations are being put into these conditions. We're seeing more shifts in allele frequencies. And then we see this apparently that the genetic variation is being maintained. So if we say, okay, we have a lot of pleiotropy, we have a lot of apparent phenotypic rate trade-offs, that gives us a potential explanation for what might be going on here, right? Because if you have lots of antagonistic pleiotropy, as a result of that, you can get essentially the maintenance of variance, because you don't want them all to fix. If they all have these costs associated with them, that will not get you to your optimal mean fitness. Alternatively, if you have things like reversals and dominance, right, you can create balancing selection as a result of this. So this is a little bit, we're playing a little bit of a game here in terms of the predictive power, but in general, it sort of fits together if we're thinking about evolution and aging. Okay, so now I'll kind of, as that backdrop, okay, what am I doing next? So looking across a lot of these results is sort of answering that question. Well, we've used this system to answer some general questions that I think are relevant or interesting to evolutionary biologists, but have we actually learned that much about aging, right? Have we done anything to improve our understanding of the molecular mechanisms that are underlying differences or observed differences in rates of senescence between these populations? And I would argue we haven't. For the most part, all we can say is the trait is highly polygenic. We can look at some Go term analysis, but usually you're just going to see, there's metabolism and neurological development. So that's not really that informative. It's not too specific. And in my experience, I think almost every experimental evolution study with fruit flies, at some point, you're going to see neurological development pop up. It just seems to be involved in everything, which makes me question how useful that is, right? So our ability to generate mechanistic insights has been pretty limited. And I think that's a problem from two reasons, right? It's one, it's like, okay, part of our goal was to learn about aging as a trait using this work, and we haven't been accomplishing that. And I think it creates a wall to even when we're trying to think about the broad patterns and dynamics, where I think their experimental evolution has been more successful than getting these mechanistic insights. But we've hit that wall where you can just have like articulate a bunch of verbal arguments for why this might be true or this might be true, but you don't actually know how the genotypes are connecting to phenotypes. So that limits your ability to make any conclusions. So thinking about ways where we could sort of get past this and potentially generate some deeper mechanistic insights, I've sort of gravitated to this approach that I think has become increasingly common, particularly in like biomedical research, where you're thinking across these different levels of biological organization. So you're thinking about, we have the genome that's, reflecting what the transcriptome is reflecting that, that's shaping what the proteome looks like, that's shaping what the metabolome looks like, and sort of going across all those layers is giving you the final phenotype that you're interested in, right? So I've done a lot of work over the past 3 or 4 years now, focusing on the metabolome. So seeing, okay, how do we incorporate this sort of metabolomic characterization into the experimental evolution framework? And will doing that actually give us any additional insights into these traits we're interested in, right? So taking this layered approach. And in the long term, the idea would be to try to approach these questions looking across these layers and trying to connect them in some form of model and seeing if that gives us just better insights. But first, I just wanted to see, like, is this actually helpful? Does this get us any better understanding, or is it just another type of data to have on the pile that doesn't really tell us anything new? So my first approach for this, I used, so this was actually done, there was another population here that are starvation resistant populations. So the C-type populations are controls for both Bs and the A-types because they were all derived from the C-types. So we just lumped them all in. So here you can see sort of the lifespan variation. So you can see the C-types are maintained on something like a 16 generation or day generation cycle, but then they're starved until 80% of them die, and then you get the next one. So, but they actually still have relatively long lifespan on average. They just happen to be very starvation resistant and stress resistant in general, right? So anyway, that's just showing, and this is a PCA based on the genetic data or the SNP frequency data from these populations. So they form very distinct clusters at that level. In terms of the metabolomics, we did targeted metabolomics of 21-day-old females. We chose 21 for this first effort just based on previous work where we know there's a lot of differential gene expression at that time. And we know, just thinking back to the demographic data, that they have these very pronounced differences in their age-specific mortality rates at that time. Lastly, I'll just talk through some of the methods of like why we made certain decisions. We use targeted metabolomics just because you can get, if you want to do quantitative work, having those more precise measurements that you can get from targeted metabolomics. So this is when you're using a mass spec to identify these compounds in a sample of tissue. So targeted, you're comparing to certain standards, so then you can kind of quantify your results, right? There's also untargeted metabolomics where you don't have those standards, so you're just kind of seeing what's there, which I think is useful for maybe broad scale characterization or absence presence. But if you actually want to do quantitative work, I think you almost have to do targeted at some level, even if you do untargeted first. Anyway, so when we have these targeted metabolomics, this time we actually, for each replicate population, we did 3 sets of 50 flies because we wanted to see how reproducible these results were. And it turns out they were very consistent across this sampling. So at least at this scale, you're getting pretty reliable measurements of what you're looking at. In this particular panel, we had 142 metabolites. And based on this PCA, you can see that the different metabolomic profiles, they are clustering very distinctly based on that, right? So it's saying this metabolome is capturing some signature of what is different between these populations. So next, we'll skip to, not sure how visible that is, but these are some of the pathways. So we did just the ANOVAs and then Tukey's to figure out where the differences are, looking at differences in metabolite abundances between these groups. And then this is showing the major pathways where or significantly different metabolites were located in. So I'm not a biochemist, so this was not super meaningful to me initially. Fortunately, as part of this work, I was collaborating with the McReynolds lab at Penn State and the Hinton lab at Vanderbilt University. And they are biochemists. They know this stuff a lot better than I do. So they looked at this data, and the first things that popped out to them were looking at the different metabolite concentrations in this TCA cycle and then in the metabolic cofactors. So when we look at things like citrate, isocitrate, et cetera, we see they're higher abundances in the A types than in the others, right? And then when we look at the metabolic cofactors, let's say if we're looking at NAD+, there are lower abundances of NAD+ than in the A types and the C types. So if we're thinking through this from, what are the actual biochemical pathways using these compounds, we would get to the TCA cycle, right? So this is when you're using citrate to produce energy, right? So the idea would be if you have you have a high abundance of citrates, it means these organisms are, getting this from their food or whatever, but they're not utilizing it. So it's going unused. Whereas in the C and S types, they're better able to utilize this, and they're, going through this TCA cycle. And usually, thinking more broadly, if you have this sort of dysregulation of the TCA cycle, this is often going to be associated with mitochondrial dysfunction. So that's giving us sort of a specific thing to go after. And I'll mention as part of this collaboration, in terms of my contributions to their work, they do a lot of work looking at how mitochondrial function changes over time in different disease states and aging in mouse and human systems, right? So we had some ideas that maybe this would be a thing in ORS, the idea being you have faster rates of mitochondrial breakdown in the A types and the C types. And just when I say mitochondrial dysfunction, we can actually see this at the morphological level, where in these aged tissues, the mitochondria are smaller, they're less interconnected, and they just don't function as well. So this is something we're following up on based on this result. So we have a collection of 7 genes that we know are associated with mitochondrial form and function. So we did some qPCRs to see how those were different, specifically between the A and C populations. So this is pretty small scale. We need to expand on this. But when we look at ATF4, overexpression of that is associated with, response to mitochondrial stress, and overexpression of MYC60 is associated with disrupted mitochondrial morphology and so on. So we're seeing some signal and this is for specific tissues. So this was a heart tissue. We're also looking specifically at brain tissue, just because we think these are major targets that might be relevant. And I don't have the results for this yet, but we're doing some work using scanning electron microscopy to actually visualize the mitochondria in this system at different time points to see if it is in fact the case that in the accelerated aging or the A-type populations, you are actually having a faster breakdown of mitochondria just on a physical level, and if that's what's happening to make them age faster, right? So that was kind of my first shot at this. When I published this work, a little while later, Daniel Promislau at University of Washington, they published a paper where they created a metabolomic clock that should theoretically be able to predict the age of a fly based on its metabolomic profile. So essentially what they did, and briefly you can see their paper if you're interested in the results, they took a handful of DGRP lines, they sampled individuals at different ages, they characterized their metabolomes, then they used elastic net regression to create a model of what metabolites are the best predictors of age, and then you can apply any data to this, use this on your data to see how well it predicts. So there's kind of a lot of interesting things going on there, right? There's like this idea that you're training it on this completely separate system that is completely unrelated, and if it actually has power to predict something useful about your system, despite that difference and differences in environment, et cetera, it means these metabolomic profiles are actually capturing some true underlying biological reality in a way that maybe other things aren't, right? Because I certainly wouldn't take the bet if someone told me to look at genetics or genomics in my populations and predict some things about another populations. I just wouldn't do it because I'm pretty sure I'd be wrong. Anyway, so we applied that to this data. So when we apply it to the C-type populations, and this is, remember, these samples were taken on day 21, on average, it does predict them to be around day 21. Whereas when we apply it to the A-type population, it predicts them to be 40 days old. So it's kind of an interesting test of this idea that this model or this approach is capturing some true underlying biological reality. And this is something we wanted to explore a little bit more deeply. And to do that, we needed to have more time points, right? I will say all of the results I'm going to be showing for the next few slides or the rest of the talk are very, very preliminary. So this is data we got back in like December. So we've only been working on it for a couple of weeks now. But I think there's some interesting findings that are emerging from it, right? Let's see, how am I doing the time? Okay, so here we're going back to the A and C populations, and we were sampling 4 time points for each, right? So in orange, we have the time points that are overlapping between them. So this is day 21, day 28, and day 35. We specifically wanted to make sure to have day 28 in there because remember, that's the reproductive or the prime reproductive window for the C types based on the selection they're maintained on. And then the others, we have these time points that are specific to a given treatment, right? So for the C types, we wanted essentially what the really old C types look like. So we collected some samples from day 70. It's not possible to do that for the A types, right? Because they're all dead. For the A types, we wanted like their reproductive window or like when selection is, focused on them, which is day 9. We also can't collect day 9 samples for the C types because not enough of them have emerged at that point because of this difference. So again, we did targeted metabolomics again. So I think the panel this time, it was a much bigger panel. So this was done in collaboration with specifically Ben Harrison at UW, and he's involved with their core. So they have, as they do more and more work, they just create more and more standards. So even their targeted panels are quite large. So I believe the total panel size for this was around 340 something metabolites, and then we had measures for 202 of those metabolites. So still not quite as good as doing untargeted in terms of doing like a full characterization of the metabolome, but getting a little better, I think. This time we only did 50 females per sample, just because we saw that consistency in the previous study across these in our separate samplings for the same population. We didn't think it was really worth it to invest in that. Instead, we wanted to focus more on time points. So anyway, here you can see PCA showing how things shake out, right? So one of the things we noticed right away is that the day 9 results, so it's circles are A, triangles are C's, and then the color gradient is corresponding to the time point, which I think is somewhat visible. Anyway, so the A types day 9 kind of cluster by themselves. And this is something we see reflected in the data, and we'll talk a little bit about. It just seems like there's a lot going on at that specific age. Part of it's, they've only been adults for a couple hours. They're also investing a huge amount of energy into reproduction, and all this stuff is ramping up. That might make it a little distinct from the other days. But we do actually see a separation between the A and C type data, with the exception of the day 70 C type data, right? So the day 70 C type data actually clusters more with the A types. Otherwise, you see this separation between the triangles and the circles. And you see some extent of clustering by days. In terms of thinking about what this means, so one of my, just going back a slide, these were kind of the 2 big questions I was asking, right? What are the metabolic differences observed between, so that's kind of the very proximate. The next is, are there distinct trajectories associated with accelerated aging, or do you just have some generic trajectory of breakdown, right? Because in my mind, that can potentially tell you something if you remember those hypotheses I talked about underlying the evolutionary theory of aging, where you have mutation accumulation, and then you have antagonistic pleiotropy. If you're thinking about the relative importance of those 2 things, if you think antagonistic pleiotropy is the big driver or major factor, it would mean because selection is actually creating differences, right? There is a distinct pattern that's better, that optimizes you for early development and reproduction. You might see very distinct metabolic or metabolomic profiles versus the C's versus, the general breakdown idea, where things are still going to be clustering by day, but slightly offset. So anyway, to do a little bit more work on this, so we used linear mixed effect models to associate metabolites that are associated with selection in time. And this is an upset plot showing a summary of those results. So, so here, oh geez. Okay, whatever. Here we have, this would be the counts for the number in each categories, whether that's selection, time, day, or the interaction. And then the vertical bars are showing you, so the first three would be the number of unique metabolites to each category. And then the other bars would be the shared overlapping metabolites based on the categories that are highlighted by the dots below, right? So one of the interesting things here is, with the exception of 15 metabolites that are unique to the time category, all of our other metabolites that are significant are associated with either selection or the interaction between day and selection. So to me, in terms of that sort of big question, this does suggest that selection is just a major force shaping these metabolic differences between these groups. And it's not, aging isn't just some genetic breakdown. It's more about what's optimal under different conditions. So from here, we've done a lot of work using some different software to, okay, how do we bin these different metabolites into different, like what pathways are they belonging to? Thinking about the correlations between metabolites and how we would expect those to look like between pathways. And looking through all of that data, we've tried to figure out, okay, what are the major patterns that are defining the difference between these groups? So I'm just going to give you a little bit of a taste of what we've found so far. We're still, in the process of really fleshing this out. But the one pattern that jumped out to us immediately was this apparent difference in glucose usage, right? And I'll come back to this. Obviously, I'm making an assumption abundance is related to usage, which may or may not be the case, but we can just make that assumption for now. So if we look at so the red are the A-type populations, the blue are the C-types, we can see the abundances of glucose in the C-types are quite low at this first day 9. So if you were to think of that as, that means they're using a lot of the glucose, and glucose is the primary sugar in their food, right? They're just using a lot of it, right? They're going through a lot of glycolysis or something like that. But then at later ages, you're seeing the abundance of glucose go up. It means maybe this specific metabolic pathway is being downregulated in some fashion. In the C-types, you see kind of a similar pattern, not quite as pronounced, and then changes at day 70, but basically there's just a lot of glucose there, and it's not apparently being broken down, or that metabolism isn't going on, right? So then we were like, okay, let's look at the actual glycolysis pathway. So this is looking at the other metabolites that are characterized in our panel that are present in this pathway. So, so you would naively expect a correspondence between the two, right? So if your glucose abundances are low, it means you're using a lot of glucose. So you should have a lot of pyruvate and you should have a lot of NADH. And we do see that at that time point. But then we notice, even as the glucose levels or the abundances are starting to go up, we still have a lot of NADH and pyruvate going on. So what's happening? How are we still having these products? Is there some sort of metabolic remodeling or a shift in what their metabolite or metabolism is prioritizing that's happening with age? So that would be sort of the speculation. And then we sort of look at that, and then we're looking at what are the other pathways that will produce NADH and pyruvate. And then there are a few amino acid pathways that will do that. So the breakdown of isoleucine, phenylalanine, and leucine will also produce NADH and pyruvate. And then if we look at the abundances of these different amino acids, we see they start at very high abundances, but then at later ages, more and more of these appear to be being used, right? So if we, again, take that naive interpretation, we're saying there's this apparent shift from sugar-based or glucose-based metabolism to amino acid metabolism that happens with age. And then the shift is happening in a faster and more extreme way in the A-type populations versus the C-type populations. And in the long run, we're trying to think through and do experiments to see, like, how can we tie this to other things we know about their phenotypes, right? So some of the big things would be egg laying and reproduction. How might that tie into different nutritional demands and what could be happening there? But like I said, this interpretation, even though it sounds kind of like a nice story, it's a very naive interpretation, right? Because the reality is, we know that observed abundances are going to be the result of the production and the consumption of these different metabolites, especially if we're thinking about metabolites that they're getting from their food. You can even add in, like, their ability to extract specific metabolites might be different, right? So it might not necessarily be as simple as saying, like, okay, this is up here and this is down there, so it means they're linked. So our next step is going to be using isotope tracing to try to confirm these results. So this is where we would essentially give them, say, tagged glucose. Then as they eat them, you can see like what molecules that glucose is going into and sort of generate those hypotheses. So sort of the pipeline I see for this work, at least that it's emerged for me, is thinking about, in an ideal world, I think you would probably want to start with untargeted metabolomics to get these broad scale differences or these presence or absence questions. And then you could potentially design panels or use large panels based on that to do targeted metabolomics. From that, you can then generate hypotheses that you will test using these tracing assays, right? As one possibility. But just as terms of a sanity check for this, we dug into the literature to see what other people were finding. And coincidentally, we actually found this paper from 2022. So this is where they use a technique where they're doing untargeted metabolomics, but they're also doing lots of tracing and looking, comparing patterns from day 3-day-old flies and 30-day-old flies to see what's different. And they actually also found a really pronounced shift in activity related to carbohydrate-based metabolism versus amino acid metabolism. The strongest patterns they saw were decreased glycolysis and more serine and purine metabolism. And then we actually looked at serine and ORS, which initially we overlooked because this is one of those that was only associated with day and not time and day or the interaction. And we actually do see that pattern where there's very high levels or high abundances of serine at earlier days, but then over time there's a big decrease in the amount of serine. So potentially some corroboration or some connection to these results. But to say anything more meaningful, I think we have to do this next step. Yes. Yeah. Yeah. So yeah, I think that's kind of what I have. I don't know if I went too fast. I was trying to plan it. I did 40 slides. Anyway, so aging is perhaps involves an increase in mitochondrial dysfunction. So that's sort of what we're seeing based on those early results. And then we're digging a little deeper into that using these microscopy approaches and potentially looking at more genes that we know are associated with mitochondrial or morphology and seeing if that's a major feature of the system. And then we have this apparent shift from carbohydrate-based metabolism to amino acid metabolism. And our next step will be following up to see if we can further corroborate this idea. But in general, the idea would be optimization for fast development and reproduction has led to just this sort of major remodeling of their metabolism that's underlying this. So I think we still have to do work connecting this to other aspects of the phenotypes, right? So I think some of the things we're thinking about are like, if you're the period where your egg production is at its peak or something like that, what might that imply about your nutritional needs versus general maintenance? If in this system, we do think they're generally optimizing around certain reproductive windows. So there's still a lot of ongoing work there. But in general, I think this, to me, it seems like a promising direction, right? That incorporating metabolomic data into the experimental or evolve and resequence framework might give us a way to get a better functional understanding of what's going on in these systems. And I think on the long term, maybe having that functional understanding might inform our ability to tease apart different dynamics we're absorbing in these systems where I think the wall is we don't know what the genes are doing, right? Yeah, so that's all I have. So this is my acknowledgements. So these are the folks that have worked in the top part, the first sets of these projects, and my more recent collaborators, and just thank all my funding sources. So happy to take any questions.